Okay, today we're going to be talking about hydrothermal vents, one of the most unique, bizarre habitats, environments that we're going to be discussing in this course. It's really, really different, obviously, of course, from what we have been talking about just last week about shallow benthic environments, which you can look at quite easily by just walking out into the shallow waters of the inland bays or see some of the same organisms in the salt marshes around Lewis. This one would be a little bit hard to walk into because it's quite deep. Um, you know, before we've been talking about environments, benthic environments that are basically in waters that are less than 50 meters. And today we're going to be talking about organisms that are found in waters that are 2,000 to 3,000 meters deep. And we're talking about these vents, these hydrothermal vents that are found at spreading zones between the plates that make up the surface of the Earth. If you think this is a little bit hard to look at, just to orientate yourself, I had to do this, but basically here's North America. We're sitting over here, and so this map illustrates the plates that make up the Earth's crust. And so this is a case where the geology of this setting is really important for understanding the basis of the life at these vents. And I think that's one of the neat things about oceanography and marine biology. We've seen already that you have to know quite a bit about physics to understand mixing and how, for example, light is attenuated in the water column. You certainly have to know a little bit about chemistry to understand nutrients and how they make up these organisms and the nutrient ratios. And here's a case where you have to know a little bit of geology to understand, again, what supports these organisms at these hydrothermal vents. So again, it was the geologists who really discovered these vents. They were predicted to exist back in the 70s. It was Jack Corliss, who at the time I believe was at Oregon State University, and he realized that given that there were these plates, and here's another picture of them showing the ridge, especially in the Atlantic Ocean, which has been studied quite a bit, should have a big impact on the seawater. And he found some potential sites for this by towing a thermistor, which is basically a way of measuring temperature around where these plates should be intersecting. And he suspected that you should see an increase in the temperature, higher temperatures, warmer water near these vents. Another star in this show is Bob Ballard. At the time was just an old-fashioned geologist. It has turned now into more of a marine archaeologist. You may have come across his name because of his role in finding the wreck of the Titanic on the ocean floor in the North Atlantic. We may see a video. I'm leaning towards showing that video in class when we meet on Tuesday. So just to remind you of what the environment looks like down deep at 2,000 meters or so. And this should be a review. There's really not much currents. Pressure, of course, is a big factor, really lots of pressure because you have 2,000 plus meters of water above you. Basically no light. For the most part, these waters are cold, cold at least for us. Really not that cold, as I've tried to argue before. It's not cold compared to what you can see even in Alaska in winter. I've been in Alaska where it was minus 20 degrees Fahrenheit. Pretty cold. You couldn't even leave a car outside without having something that kept the engine block warm and expect it to be able to start the next morning. And then, of course, the salinity is full strength, and there's plenty of oxygen. Oxygen is not an issue there. The big difference that we're going to see is temperature and some of the other properties that vary and are quite different at vents. And so to remind you, in everywhere else in the ocean, in the deep benthic environments, it's supported by food that's raining down from the surface. All that food is made by prime producers, the phytoplankton that are at the surface where there's light. And these particles sink down. They're either sometimes the phytoplankton themselves will sink, at the end of a spring bloom. You also get fecal pellets from the zooplankton feeding on the phytoplankton. You get occasionally a whale that dies, and or a larger organisms, a fish that dies, and somehow makes its way down to the bottom. And that flux decreases exponentially. This should be all familiar to you already. It decreases exponentially. Again, I don't want you to remember this equation. That's the equation that folks have used. What is important to know that it decreases exponentially with depth. And that just comes from the fact that there's basically a fraction of the material is taken out per depth as it goes from the surface to the bottom. And that's when you have a constant fraction with that depth, you end up with an exponential type of decay. So the end result is that there's very little of the material that's at the surface will eventually make it down to the deep benthic environments. And so 1%, that's just, that will change, of course, depending on how deep you are. And so, but that's just kind of an order of magnitude. If you were forced to pick one number without knowing anything and just told it's deep, 1% is not a bad guess. So there's something. So here's what it looks like. So we talked about the deep benthic environment being very diverse, but the other main characteristic of these places is that it's very sparse. There's not many organisms. The biomass is very low. So I wanted you to keep in mind this picture, basically a desert of very few organisms. And of course it's a desert because there's very little organic matter that makes its way down to the bottom. There's very little food for these organisms. So hence there's not many of them. They're diverse, but there's not many of them. So that changed when folks started to look at these vents. And a big part of the story is this submersible, the Alvin. Turns out that Alvin was named actually after a person that was kind of the main instigator who wanted to get one of these submersibles to look at various processes. And his name was Alvin, not Alan Vine. So they kind of corrupted or changed his first name to put a V in it, and got Alvin. And it just was at the same time there was a cartoon about a chipmunk that you perhaps have seen. It was kind of popular when I was a kid, and so that kind of helped or whatever. It was another kind of little inside joke about naming it Alvin. I also learned that one of the first ships to help carry Alvin around, now it's the Atlantis 2 that carries Alvin from one place to the other. But originally it was Lulu. It was a ship called Lulu, and that turned out to be Vine's mother's name. So anyway, it was built in 1964, but it's been renovated many times, most recently in 2013. So there's probably not one single piece of metal on it that's from the original 1964 version. So this is about 23 feet long or so, 12 feet high. As I mentioned before, it can fit only 2 scientists and 1 pilot. One of the many joys of being on it, as I've said, I've never been on it, my wife's been on it, is that there's no facilities, and so you have to kind of hold it for 6 hours or so while you're on the Alvin. So when Alvin was taken down by the geologists, and Ballard was on one of the first dives to one of these hydrothermal vents. This is what they saw out the window. And you can imagine, after seeing nothing and nothing, just a desert of no organisms at all, it was a tremendous surprise and shock to see this ride of life at these vents. And now we know what these are, and we know quite a bit about their biology. These are tubeworms, and we're going to hear more about them in a minute. So these organisms are found at vents, and so vents are at the joints between the tectonic plates. What's happening is that these plates are basically spreading. They're moving apart, and as a result, they're leaving a bit of a gap. And through that gap, the molten magma, can come up. And it's not actually the magma itself that will reach the surface, that's not that important for our part of the story here, although there are eruptions at the bottom of the ocean. What's more important is that seawater can get down to be heated up through these cracks and comes back out to the bottom part of the ocean. So before we get to that, here's just a little bit clearer map of where these vents have been looked at. One of the more famous ones is the Galapagos one, and then we'll talk a little bit more about and just one picture about the vents, which are very different, that are found along the Mid-Atlantic Ridge in the North Atlantic Ocean. But there are others found that have been looked at least, other places in the oceans. The other one that's been looked at quite a bit is those off of Oregon and Washington. So here's a really complicated diagram showing all the things that are happening. And so this, if for those of you who know more about the chemistry, I encourage you to look at this. But here's a bit simpler diagram, and I think has the most important points that I would like you to focus on. So again, what's happening is that seawater can go through just kind of by diffusion, get through the rocks and get down into the hotter parts of the mantle. So that's probably happening all over the place. But what's different at the vents are these cracks where the plates are spreading that allows that seawater to come back up. And so what's happening as the seawater permeates down into these hot rocks, it's heated, of course. And what happens is that everything's reduced. And in particular, the sulfate that's in seawater, sulfate is the dominant sulfur compound that's found in seawater. It's reduced down to hydrogen sulfide. And you maybe remember hydrogen sulfide is that poison that's made by, normally in salt marshes, for example, it's made by organisms, but here it's made by geology. The hot rocks heat up the sulfate and make hydrogen sulfide. And then this hydrogen sulfide comes streaming back up through the vents. Hydrogen sulfide and a bunch of other reduced compounds. And this diagram has many more of these reduced compounds that come back up through the vents. So that's we'll find out is the source of the energy that fuels this whole ecosystem. So these reduced compounds have energy that organisms are able to harness and make a living off of. And that's what supports the whole ecosystems. So what's very characteristic of these vents often is the fact that you get these chimneys, these big towers of metal sulfides that have precipitate at the event. And what's happening as hot water comes out really 350 degrees Celsius, 400, 500 degrees Fahrenheit, comes out and hits the 2-degree water of seawater, and it of course it cools. And while these reduced metals can be soluble at really, really high temperatures, they aren't soluble at the low temperatures, and they fall out of solution. They precipitate, and they form these chimney-like structures that are indicated here. So the question is, why? How can you get something that? So back up. What's the boiling point of water in Celsius? Well, at atmospheric pressure, of course, it's 100 degrees Celsius. So here we're seeing water that gets up to 350 degrees. And so how can that be? How can liquid water stay liquid at 350 degrees Celsius? Well, the answer, of course, is that the pressure is tremendous. This is basically a pressure cooker that's keeping the water liquid at that high temperature. And as that water comes out again, it's all the things in it, many of the things that were soluble at the very high temperatures become insoluble and fall out of solution, and you see the precipitation of those metals and forming the chimney. So here are a few videos that we're going to look at. We're going to look at one of these, and as I mentioned, I'm leaning towards showing the Ballard and Bill Nye video. He's a very engaging speaker. And so we'll probably look at this one, although the quality of the video is not as good as some of these other ones. So we'll do that in class on Tuesday. So just to talk a little bit about some of the organisms that we see at these vents. So these vents come and go. they have a finite lifespan, and so there's a somewhat we know somewhat about what happens when a new vent appears, and we see these one type of tube worms. I'm not going to ask you to remember that genus name, but I'm going to ask you to remember the dominant tube worm that we see at vents, Riftia. But so anyway, there's this succession of organisms that occur at new vents that we know a little bit about. It's hard to study because you can't stay down there for weeks on end. Certainly, you can't send a sub down every day for months at a time. You can only look at these things from one year to another, and so that's how we know there is some change in the community over time. So most of the work has been done on these vents in the Pacific Ocean. The North Atlantic vents and along the Mid-Atlantic Ridge are quite different. They have a different chemistry. They have carbonate rather than the iron sulfide that dominates the vents in the Pacific Ocean. Why that is probably due to the geology of these places, and so. But suffice it to say, there's big differences. So these vents don't have high amounts of iron and other metals. They're more alkaline, and that becomes important when we talk at the very end about perhaps the origin of life at these vents. So anyway, they differ. And I just want you to give I just want to give you an impression or overview of these hydrothermal vents and some of the chimneys that we see there, and then we'll see in a minute the organisms. They're not all the same. They all share the characteristic, though, of very hot water coming out with reduced compounds that are supporting life there. So they're called, the ones at the Mid-Atlantic Ridge are called the Lost City, and I guess they got that name because of these white towers of carbonate that form rather than the black towers from the iron sulfides in the for example, the Galapagos Ridge vent. So another worm that's seen there is Alvinella, and why this is important is because it seems to live at really, really high temperatures. So this is a kind of a range of some of the temperatures that are thought to limit life. The winner so far is archaea. Remember, archaea look like bacteria, but they're a totally different domain of life. They're as different from bacteria as they are from eukaryotes, that is us. So they're the winners so far, found, I believe. So again, that's above boiling point of water. So it has to be at fairly high temperatures, fairly high pressures to have that water be still liquid. And so anyway, you can see some of the other winners in terms of temperature and how hot they can stand. And so this Pompeii worm, the Alvinella, it can withstand apparently really, really high temperatures for a metazoan, much more so than say for some of these terrestrial organisms that are in the desert. So here's another crab that's seen at the vent. It's not that important, perhaps. I'm just mentioning it because there's a Delaware connection to this. These three people are all from Delaware. Chuck Baffanio is a professor here who just recently retired. Gina got a master's degree here, and now she works at NOAA, and that's my wife there. She just retired, was a biologist who was working on some of these animals. And what's really striking, I still remember very, very vividly, picking some of these folks up, my wife of course, and it was I think Christmas Eve, and they had all these really cute little crabs with them. They had brought them back from the vents, and here's what it looked like, and it's kind of a red Christmas-like color. And they tried to keep them alive to watch it metamorphose into the adult, and they were able to keep it alive, but it never really completed its metamorphosis. And the reason why this paper was published in Nature, which is of course a really, really high-profile journal, is because of the story about how it started off with eyes, but over time it basically lost its eyes, and because the adult doesn't need them at the vents. There's no light down there. So again, I want to remind you that all this life is supported by energy that's coming in the form of these inorganic compounds, and the most important one is hydrogen sulfide. And so you definitely need to know that. There's a 2 there, hydrogen sulfide. So how does that work? Now, you should know the answer for this already, because we just talked about it, that there's some organisms, even in shallow benthic environments, even in salt marshes, that do this, that they use, take advantage of bacteria that are symbiotic with these some types of clams, for example, that oxidize hydrogen sulfide, and they make energy from this, and they use that energy to make more of their own cells, and then that organic matter also goes to feed the clam, in the case of salt marshes, or the hydrothermal vent tubeworm in the case of the vent organisms. So whereas the hydrogen sulfide in a salt marsh comes from sulfate reduction, in the vent it comes from geology. Remember how that hydrogen sulfide comes from the seawater that was superheated by the hot rocks that reduced this sulfate to hydrogen sulfide. So that's where the energy comes from. So this equation just gives 2 compounds: the sulfate, which is the dominant form of sulfur in the oceans, in oxic oceans. But also there are many other intermediates in sulfur chemistry. Sulfur chemistry is really, really difficult and complicated. And we're not going to get into it. You'll be happy to hear. The things that I want you to remember are hydrogen sulfide, and at the other extreme is sulfate, which is the oxidized form of sulfur that we see in seawater. So how does this work? Well, let's start off with what we are doing right now. I hope you're doing this right now. I hope you're breathing, right? Even though you may be sleeping, you're still breathing. And what you're doing is you're oxidizing organic matter. Remember, CH2O is a kind of shorthand way of talking about organic compounds. And what you're doing is you're taking that organic material and you're using oxygen, of course, and you're producing CO2 and some water. And you're getting energy from this. You know, what's not given in these equations is you're getting energy. Not writing out really clearly there, but I hope you can, well, at least hear me say energy if that's not real clear. Well, I don't know why it's not clear. Anyway, so you get energy. So let's generalize and take this equation apart to see what's actually happening. So what's happening in terms of chemoautolithotrophy is that the organic material is being replaced by an inorganic compound, another electron donor. And so let's take that apart and understand, what do I mean by electron donor. So in aerobic respiration, which is what you're doing right now, you're taking that organic material, and that organic material is being oxidized. It's giving up its electrons, and then those electrons are going to oxygen. So the electron donor is organic material. An electron acceptor is oxygen. So it's the donor is giving up the electrons, and obviously the donor, that's the word we use, and the receptor in this case is oxygen. So what the chemoautotrophic organisms do is that the donor now is hydrogen sulfide, but the electron acceptor, not receptor, it's acceptor, is still oxygen. Now we won't go into variations on the acceptor part, but for this part of for this reaction and for the story about the vent animals, oxygen is the most important electron acceptor, and it turns out it is the most important electron acceptor in all all habitats. So again, hydrogen sulfide is analogous to organic material. Both are electron donors. They're being oxidized. They're giving up their electrons, and those electrons are going to oxygen. That is the electron acceptor. And so in the process of doing that, energy is released, and that's what's being harvested and used by these organisms to make biomass. So this is only done by bacteria and actually archaea, No metazoan is able to do this. No eukaryote is able to do this. So again, energy is being released by the bacteria. So I just want to back up. I have it going only to elemental sulfur. I was being a little bit lazy. These organisms actually take it all. Some organisms will do it, only take it to elemental sulfur, but these organisms actually take it all the way. The ones that we're interested in here today take it all the way to sulfate, all the way to the most oxidized form of sulfur. So again, the most important thing is that they're getting energy from that. And that energy is used by the bacteria. So again, the hydrogen sulfide comes from the vents. The oxygen comes from the surrounding water. So the water that the vent fluid is going into has lots of oxygen. Remember, oxygen is not a problem here. And ultimately, that oxygen comes from the phytoplankton at the surface, and also half of it comes from terrestrial plants as well. So the question is, how much energy do you get from doing this? And I mentioned this already on Thursday. You don't see large organisms running around doing this type of metabolism. As I said, only the bacteria and archaea are able to do it. And the reason why is because you don't get a lot of bang for your buck for doing this. Here are the energetic yields for various types of chemoautolithotrophy. Here's the one we just talked about. The oxidation of hydrogen sulfide. This is actually at seawater pH. That's what hydrogen sulfide really looks like. And you can see the energetic yield here. No, these numbers are not important for you to know and certainly memorize. This is what you should remember and know. That is, the oxidation of inorganic compounds is just not a great way to make a living. If someone gave you a choice of running on inorganic compounds, I suppose it'd be somewhat advantage. You won't gain much weight, and I suppose you could just have a little tank of hydrogen sulfide on your back rather than having to eat. But overall, not a good way of going. Very much less energy released by this. So to put these numbers in perspective, here's what you get from oxidizing glucose. So you can see there's just order of magnitude difference in terms of using hydrogen sulfide compared to oxidizing glucose. So stick with glucose. So now the question is, if you look at the equations that we've been talking about so far with hydrogen sulfide, there's no carbon. There's no carbon. Of course, when we oxidize glucose and use glucose or organic matter more generally speaking, there's plenty of carbon. We get our carbon from the compounds we ingest. With a chemoautolithotroph, they have this problem. Where do they get their carbon? And the answer is CO2. So these organisms are really autotrophs. They are autotrophs also. And so their full name, if you want to get really formal about it, is a chemoautolithotroph, or sometimes chemoautolithotrophes. And that describes everything that they do. The litho part is Greek for rocks, and so they're getting their energy from rocks. They're getting their energy from these inorganic compounds. The auto refers to the fact they're making their own food, making their own carbon, and they're getting their carbon from CO2. But in the symbiotic bacteria world, when people talk about these organisms, they'll use the term chemoautotrophs or chemosynthesis, which I hate, but that's what they do. And the reason why I hate that is because basically we are doing this as well. We are chemoautotrophs. We exist on chemical energy, unlike plants, which exist on phototrophy and sunlight. And we also make our organic material by chemosynthesis. So that's why I think those are not good terms. But in fact, that's what the people do. So if someone tells you that they're working on chemotrophic symbionts, you should know that they're really working on these type of organisms that are chemoautolithotrophes. Okay, enough of my rant. So what you should know now is that hydrogen sulfide is the energy source for all the life that we see. A huge ride of life at hydrothermal vents. That is used by these chemoautolithotrophic bacteria for energy, and you get their carbon from CO2. And so these bacteria are the ones that feed the rest of the organisms. They are the base of the food chain at the vents. And you should know that the vents are at these cracks between the plates. And so what happens is that the seawater seeps into the crust. It gets really hot, and while it's getting heated, all those otherwise oxidized compounds are reduced. And the most important one for our story, at least here, is the reduction of sulfate to hydrogen sulfide, and that hydrogen sulfide again becomes the energy that supports the whole ecosystem there. So that those organisms can be just free living. They can form mats near the vents, and that's kind of indicated here by this kind of whitish stuff that's growing around the vents. And there's organisms that will feed off of these mats. So this is not like the deposit feeders. These are mats that are growing on rocks, and so they may take in a bit of rock material, but they're basically scraping it off the rocks and getting whatever organisms that may float off. But the really more really fascinating part of the story, I don't know what's more fascinating, but this is certainly a really great part of the story, is that there are many organisms at the vents that have symbionts. That basically these chemoautotrophic bacteria are living inside of these vent animals. They're endosymbionts. So they're living within the tissue of the larger organism. So you can see here, that's what's illustrated here. So here's the membrane of the host, and inside of it, it has all these bacteria that are the symbionts. So and so again, these organisms need the sulfide, the hydrogen sulfide. They need oxygen, and they need CO2. So the most spectacular example, and what Ballard saw out of the window of Alvin when he was cruising around near the first vent, were these tubeworms, Rifty. I'd like you to remember that genus name, because that's kind of a famous one for looking at these organisms. They really originally were put into their own phylum, Vestimentifera, but more recently they've been put back into the annelid phylum with all the other worms, much more boring earthworms, boring I guess in terms of not as exciting. So anyway, that's their taxonomy. But what makes them really different, and there are relatives of these much smaller, more not nearly as spectacular versions of them in shallow waters. And zoologists already knew before the vent animals were discovered is that they basically don't have much of a digestive tract. They don't have anything. They don't have a mouth. They don't have an anus. And so, of course, it always was a mystery how in the heck they could survive. The other thing that was really interesting about these organisms when they were first discovered is that they were the trophosome is just a term that's used to describe the body cavity of these organisms, and it was loaded with sulfur granules. And so I actually mentioned that that the organisms produce. I don't know why it's not working better. Let's see if my finger works better. Yeah, my finger works better. Produces hydrogen sulfide, but these organisms also will the bacteria will oxidize the hydrogen sulfide only to sulfur, elemental sulfur. So that's what these organisms were first discovered to have. So these granules weren't really separate from the sulfur granules. I mean, so granules is just a word to describe particles. Another just a nondescript word to when you don't know what it is, you call it a granule rather than a particle. I'm not sure why you use one word or the other, but the zoologists were using that word granule to talk about these things that they didn't know what they were. Here's what they look like, and they look like little globs. Now we know that they're bacteria, but when the zoologists were looking at these things, they didn't know the heck they were looking at because, of course, they were zoologists, not microbiologists. And it took a graduate student who knew about bacteria to really figure out what was going on. So the first author on this paper at the time was Colleen Cavanaugh. Well, even now she's Colleen Cavanaugh, but at the time she was a graduate student. And these two guys were famous microbiologists, and these two were the zoologists. But it took the graduate student to figure out what was going on. And she, as I said, she was a graduate student, and she heard a lecture by these. I think he was the second author on that paper that I just pointed out, and the paper was published in Science, which is of course a really high visible journal, and it was, you know, it warranted being published in a high visible journal because it was such a really great discovery, a first discovery of these bacteria, these chemoautolithotrophic bacteria being inside of an animal. And as I said, she heard it while hearing a lecture by Meredith Jones and came up with the idea. And she had a real battle to get others to believe her. And Janusz, you know, didn't believe it actually, even though he's a microbiologist and should have had bacteria on the brain. He didn't really buy it. And Colleen had a really tough time convincing these people. You know, she had two strikes against her. First of all, she was a graduate student, so you know, students don't know as much about things as professors, right? Well, in this case, she did. And the second thing, I think she was a woman, and at the time, back in the late 70s, early 80s, things were even tougher for women than they are now. They're still not perfect, and we have a long ways to go. And I have to say, I think Janusz was originally from Germany. His first name at H is Holger. I always found him to be a bit intimidating. He's since passed away. You know, I guess maybe that German accent. I just kind of, you know, seen too many movies, and just have this stereotype about with that German accent that kind of makes him a bit more formidable. So anyway, she had a big, many problems getting accepted to and convincing people that this was actually what was going on. But in the end, you know, she did pretty well. She's now been a professor at Harvard for many years. So anyway, that's a little bit of the backstory about how these things were discovered. So let's go back to the biology and talk a little bit about, you know, this being a symbiotic relationship. Who gets what? So what does the animal get? Well, you should know. So it's getting food from the bacteria. It's basically getting some material that's coming from living cells. Some cells must die at times, and so the animal digests those dead cells. It perhaps even eats one or two living cells. It needs to keep the living cells a bit in check. The population can't get too large, and so it has to eat some living cells to keep it under control. Now, the other thing that the animal gets, it detoxifies this poison. Remember, hydrogen sulfide is really toxic to many organisms. And so one mechanism that some organisms use is actually having these bacteria. So that's an advantage to get rid of the H2S, rather than dying from it. So what does the bacteria get from it all? Well, they get a steady supply of food. Their food, I mean, I shouldn't use that word food, and you shouldn't either. It's not nutrient either. In this class, nutrients are things like nitrate and phosphate. This is an energy source for these bacteria. Oxygen, what's oxygen here? It needs oxygen, right? It's an electron acceptor for these bacteria. Also, we can't forget about top-down factors. Some protection from grazers, although there's, it's eaten by its host, but perhaps less so than if it were out there in the wild world. So a little bit more detail about what's happening here. Bob Ballard in the video that we probably will see by now, if this is class, talks about landing on some of these tubeworms, which is horrible to think about. They definitely don't try it. They don't do that now. And mentions about blood coming out, or that's what I think the word he uses. And it really was blood. It was really the blood that we see in these tubeworms is really similar to our own, in a sense it's something very similar to hemoglobin, the main thing that gives blood its color. And it gives its color because of the iron that's in it that binds oxygen. And so the hemoglobin in the tubeworm blood binds oxygen, but also binds H2S, and that's the reason why H2S is poisonous. So again, at seawater pH, it's HS. So at seawater pH, basically it goes back and forth. It's mainly as H minus sulfide. for reasons we don't need to get into. Anyway, so the hydrogen sulfide binds to the hemoglobin, and that's why it's toxic to these organisms, to other organisms, but it's good for the tubeworm because it brings the hydrogen sulfide to the bacteria, and it's able to carry out the oxidation of that hydrogen sulfide and releases energy. And then the organic material from that, some of the organic material goes to feed the tubeworm. So it turns out there's other, we mentioned this already, that other organisms have this type of symbiotic relationship, and they have the hemoglobin as well. And then I think what's really neat is that there are still other invertebrates, snails, that have a different type of it's not hemoglobin, but it's probably, I don't know if they call it cyanoglobin, but it has blue blood. That is probably copper. It is copper. That's the metal that takes on electrons and takes on the oxygen and hydrogen sulfide. Okay, so I think it's useful to compare photosynthesis and chemoautotrophy, and compare the more typical ecosystem that we are familiar with and what we've been talking about so far in this course with the one that we see at hydrothermal vents. And so if we talk about the energy, that's where things are really, really different. Of course, for all the systems that we've been talking about so far, it's ultimately the sun. It's sunlight that support either plant growth on land or phytoplankton growth in the oceans. And then eventually that goes to support the rest of the food chain. But in synthetic based systems, that energy comes from chemicals. It comes from hydrogen sulfide. And unlike the prime producers that we see on land or in the rest of the ocean, the prime producers for these systems are sulfide oxidizing bacteria. So these organisms are either free living or they're found in the tubeworms as symbioses. But after that, basically, there's a huge difference in prime producers between the two systems. It turns out the way in which carbon is actually fixed, by that I mean going from CO2 to organic material, is exactly the same. Is exactly the same as the Kelvin-Benson cycle. So if you memorize that for some course for thinking about plant or algal photosynthesis, the same cycle applies to these hydrogen sulfide oxidizing bacteria. And then, of course, the way in which the system is supported by these organisms is analogous. You can't say it's the same. There's certainly a huge difference between what happens with insects on land and some of these invertebrates in the oceans, but it's totally analogous in terms of the fate of carbon after that. Okay, so why are they so unique? Again, it's the bacteria, these chemosynthetic bacteria, the prime producers, not the land plants or phytoplankton. And the source of energy is hydrogen sulfide, that is a reduced inorganic compound, not light. There are other forms of chemoautolithotrophy. That one table kind of listed a few of them. They're really important in terms of various biogeochemical cycles. But in terms of supporting ecosystems, hydrogen sulfide is king. And it's mainly because there's just so much of it. There's just so much of it coming out of those vent fluids. But they're, the biology that we see at the vents are unique because, one, the bacteria, the prime producers, not the land plants or phytoplankton, and two, the source of energy is really different. And again, what's also really unique is the key role of symbiotic bacteria. So as I think about that, symbiotic bacteria are found in all organisms, including us. And so in some ways that's not as unique as this implies, but it's probably true to say that that the hydrothermal vent, certainly the Riftia, the tubeworm, is they can't survive without the symbiotic bacteria. We can't either. Yeah, but these the source of energy for these organisms they're totally dependent on these symbiotic bacteria. Okay, just a couple final topics before we quit. And one topic is basically thinking a little bit about how new vents are colonized. So I mentioned the fact that these vents do not last forever. They may last years, not decades. And then there are cases where whole research projects have been based on going back to a particular vent field, and the scientists go back there and discover it's all dead. The vent is turned off, and so they're out of luck. So the question then becomes is how do the new ones start up? Well, of course, that's a question in geology, but also the question in terms of biology is how are they colonized? And so the only answer can be is that the larvae from one vent has to go and swim around and just get lucky. There has been some thought that maybe whale falls and other sources of organic material, which can give rise to hydrogen sulfide production, serves as kind of stepping stones between the vents. I don't think there's much support for that. And generally it seems that it's really hard to go from one vent to another. And I have this map up here because it basically gives you a bit of an idea about the distances. Of course, we're talking about thousands of kilometers in some cases, separating one vent from another. So and we can look at that gene flow by genetic methods and see that there's not a lot of it. It's really low exchange of genes between sites, and often you just see species endemic for one site, so really unique from one site to another. So this is kind of stuck in here. I think we've already talked about the fact that we see this type of symbiosis in other places, and one that we've already mentioned is a salt marsh bivalve, the Solomaya. Colleen actually worked on that as well after she got done with the vents, and then she actually also worked on the methotrophic bacteria that we talked about already for the Gulf of Mexico. So the final topic I would like to talk just a few minutes about is the origin of life. Huge topic, obviously. So to back up, I was thinking, why should we be interested in hydrothermal vents? I often like to begin, well, who cares? Why, as budding marine biologists, you should learn about these habitats and these organisms. We've seen various reasons, because we eat the food, or someone else eats it, or other reasons. And at first I was thinking, well, hydrothermal vents, I'm not sure if they suddenly would turn off, would life be really that different for those of us on the surface and on land? Probably not. Maybe a geologist would argue with that. A geochemist would say something about how vents are really important in geochemical cycles. But yeah, I'm not sure that we would notice a difference. But so why have a whole lecture about hydrothermal vents? Well, I hope you appreciate that it's really exotic, neat, if nothing else, environment, and neat set of organisms. Totally different, having these vent worms supported by endosymbiotic chemoautolithotrophic bacteria. Totally different, right? That should be enough reason. Another reason is perhaps it explains how we came about and how life all started. There's some hypotheses that life started at a hydrothermal vent, that the first cell was actually at a vent. And so the arguments, I'm not going to go through all of the arguments. I have just two slides about this. So one argument is basically that you have a source of energy, that is a reduced sulfur. Also, we know that the surface of the Earth, when it first formed 4.5 billion years ago, was a pretty hostile place. You wouldn't want to try to take a vacation on the surface of the Earth at 4.5 billion years ago, because you'd probably die real quickly. First of all, there wasn't any oxygen, but even if there were oxygen, you'd be probably bombarded by various things coming from space, and you probably would die of cancer real quickly, because the UV light was really, really strong. There wasn't any ozone or much of an atmosphere built up to protect us from UV light. So it was really harsh, not only for us, but also for any life form that was trying to evolve. So of course, at a vent, you don't have that problem. You're kind of protected against all that, all those things coming from space that would kill you. So also at vents and is you have a high concentrations of all the other things that are necessary for life. So the other arguments is that the first cell probably was a chemoautotroph that existed on inorganic compounds. The chemistry is relatively simple. It had, it seems pretty clear it has to be an autotroph. Maybe you can make the argument that somehow a primitive heterotroph was living off of the organic compounds that had to be there first. But most evidence points towards the first cell being an autotroph. The other thing about vents is that there's all sorts of these weird elements that are found there in really high abundance. And some of the enzymes that we think are rather old, the nitrogen-fixing enzyme nitrogenase is one example. And some forms of nitrogenase have some really weird elements like vanadium and titanium that are really abundant at vents. Elsewhere in the oceans, you don't see those elements in high abundance, but at vents, you don't have to worry about them. And then finally, I think that in some ways the most neat part of the whole story is the fact that that it seems that the organisms that really like high temperatures, the thermophiles, and then the hyperthermophiles, those that really, really like it hot, seem to be near the start of where life started. They're at the base of the tree of life, and we'll see that in a minute. I'll give an example about that in a minute, the last slide for this lecture. And here are a couple papers about this story. One just appeared a few weeks ago, actually. And you can see astrobiology. So there's lots of connections between work looking at the origin of life and work looking at the possible existence of life on other planets, perhaps on the moons of Jupiter, like Europa. And this paper right here makes the argument that life started at an alkaline vent like that seen at the hydrothermal vents in the North Atlantic at the Mid-Atlantic Ridge that I talked about before, the Lost City vents. So getting back to this piece of evidence that the thermophiles, hyperthermophiles, are found at the base of the tree of life. Now here's one of the more complicated trees of life that we've talked about so far. So just to get you orientated, so we're up here with all the other animals, just a little branch of with all the chordates are indicated there. So here are the rest of the eukaryotes. Here are the bacteria right there. And you can see that these organisms that have the thick lines there all are hyperthermophiles. They're all able to withstand really high temperatures, as much as higher than 80 degrees Celsius, near the boiling point of water at atmospheric pressure. And also the archaea that are known to like high temperatures are also near the base of the tree of life. So this is the base, of course. We don't know exactly what that cell is. Of course, that's a $10,000 question or whatever. But by constructing these trees, you can see that these organisms are the most deeply branching. They are closest to that first cell. And so they all turn out to be hypothermophiles. So you can see hypothermophiles and thermophiles in hot springs, such as at Yellowstone National Park. But the most hyper of all the hyperthermophiles are found at hydrothermal vents. So that's one reason, besides just one reason to look at hydrothermal vents. But I think the most best reason is that they're just such fascinating organisms and such unique organisms and unique environments. So with that, I'll quit and we'll move on to our next lecture next Thursday, which will be about another set of really interesting organisms. The organisms found in the mesopelagic zone. And I'm trying to stop this. There we go.